Hello, everyone. Welcome to Pediatric Grand Rounds. It's my great pleasure today to introduce Dr. Nikki Bush, who is the Lisa and John Pritzker Distinguished Professor of Developmental and Behavioral Health, and she is the chief of our Division of Developmental Medicine in the Department of Pediatrics. She's also in the Department of Psychiatry and Behavioral Sciences. Dr. Bush has devoted her career to research and clinical care, focused in particular on underrepresented communities who are experiencing high level of adversity. Her research is really focused on the associations between social determinants of health and biology, and how that impacts on mental and physical health, and the ways in which environment shapes development. Dr. Bush has a long track record of extramural funding, including from the NIH, and most recently, she was awarded a California Office of the Surgeon General's ACEs in Precision Medicine Award. She's also a passionate advocate for advancing DEI in academia and public health, and has been recognized for her efforts in this area in many ways, including most recently receiving the UCSF Chancellor's Award for the Advancement of Women. So it's a great pleasure to have Dr. Bush with us today. And I will hand it over to you and look forward to your presentation.
Thank you, Rafi, for that introduction. It's a real privilege to be here among my pediatric colleagues and to get a chance to share some of the research that we've been doing across UCSF and around the country. So in terms of disclosures, I have no financial relationships to disclose, but I do want to acknowledge that I reside and conduct much of this work in the San Francisco Bay Area, which is the unceded ancestral territory of the Ohlone people. I'm going to talk today about how adult problems tomorrow depend in large part on how children grow up today. You know that, or in pediatrics, but I'm going to argue that it's even before they start to grow up that we need to start thinking about the development of health and disease. My research is focused on understanding how individual differences in temperament, genetics, and stress reactivity across a variety of biological systems interact with the range of environments that humans are embedded within: their homes, their families, their classrooms, their neighborhoods, the toxins in the air, and how these interactions help us understand who's most at risk for health outcomes. However, rather than focusing on reversing negative outcomes in adulthood, we need to really increase our focus on the developmental processes that occur much earlier in life rather than waiting for disease to manifest. So we really need to push health and disease out and really think about development. And we have to consider the interactions between individual differences in genomics and stress reactivity at the beginning of life and their interaction with the environment, and how those actually shape subsequent individual differences in biology over time. And that biological embedding of experience, of social experience, accounts for the shaping of individual stress reactivity and other biological systems iteratively over time and throughout development. These phenomena occur throughout the life course, but they're most pronounced and the effects are greatest of social environment early in life. And it's really important to also keep in mind that the socioeconomic status shapes those opportunities and experiences, very much influencing the privilege or the disadvantage that we are exposed to in those environments, and subsequently our biology. One type of social experience that's gotten a lot of attention recently is collectively referred to as ACEs, or adverse childhood experiences. Most of you are quite familiar with this by now. Importantly, this has been a key focus of research for hundreds of years actually within psychology, and it's been great to see pediatrics embrace this. It was popularized and a lot of attention was brought to it in the late 90s with Folletti and colleagues, who did longitudinal work to really demonstrate that not only are ACEs common, with nearly 2/3 of adults having at least one by the time they reach adulthood, but that this has major impact on a variety of chronic diseases, morbidity, and mortality across the life course. Indeed, there's been a huge bolus of research that has demonstrated that ACEs have long-lasting effects on health. You name your outcome, there's typically, unless it's a rare genetic disorder, and even then it's often amplified by social experience related to adversity, impact on obesity, diabetes, depression, suicide attempts, STDs, heart disease, cancer, stroke, etc., but also health behaviors and life potential, including graduation rates, incarceration risk, etc. So this is a really potent social effect. But importantly, I think there's less of a conscious awareness of the social determinants of health of socioeconomic status, which is actually a much more profound and impact over time on the development and health of individuals. And in particular, these begin early and in life. At conception, poor children are 7 times more likely to be important for our health than children in the highest income families. And children whose parents have less than high school education are 6 times as likely to be important for health relative to those with a college degree. And importantly, that this occurs within racial or ethnic classes. So this is amplified by intersectionality, but is not dependent upon it. So even within African Americans, you see this very standard graded association between income, education, and health that we need to be really mindful of. So I wanted to understand, how does this get under the skin to affect health? What are the biological mechanisms? How can we identify early risk for this path towards disease? And how early can we discover these things? How early can we detect these risks? And so I became very interested in the biological embedding in early childhood. I joined UCSF back in 2007 and had the great privilege of working with Dr. Tom Boyce and Nancy Adler, 2 gurus and leaders in their respective realms. And I worked on their community sample called the Peers and Wellness Study, which enrolled 338 children in kindergarten from the East Bay area. We followed them through age 8. It was a very diverse sample, quite representative of the East Bay area. And makes it kind of interesting to interpret the findings, because these could be your neighbors or your children. One of the first things I was interested in was understanding whether we could detect effects. We had seen some SES and HPA axis function evidence in adulthood, but I wanted to know how early we could detect these differences. And so one of our early papers really focused on this. And we took measures of children's daily cortisol output, looking at their cortisol levels at the beginning of the day and end of the day across 3 different days, trying to get a general robust measure of their general output. And we found that family adversity and poverty uniquely predicted heightened cortisol output in kindergarten children. This was some of the first evidence at this age, at the time. And what we also found, not only did adversity in the family have a graded linear association, such that the greater the depression, anger in the home, restrictive parenting, marital conflict, financial stress, and overload in the family with roles and responsibilities, as those adversities increased, the children's chronic daily cortisol output also increased. Interesting, we found a curvilinear association with socioeconomic status in terms of income and education, such that the wealthiest kids had a moderately higher cortisol daily average, whereas the children who were the most impoverished had a quite high daily output. And what we think this curvilinear representation suggests is that children who enter school actually want a little bit of cortisol to optimize their learning and their responsiveness, and so that this may be a benefit to those in the wealthiest families, or it may represent pressure due to more constrictive school settings. But either way, the take-home point of this is that both income and education, as well as social adversity in the home, is related to allostatic load in 5-year-olds. We also found the same type of story using autonomic nervous system measures in children 5 years old. We hooked them up to EKGs and exposed them to a series of developmentally appropriate challenges, assessing both parasympathetic and sympathetic reactivity, as well as integrated heart rate. And we did this multiple times across the kindergarten year. And what we found is that family level objective social status, so income and education, was associated with longitudinal changes in children's sympathetic reactivity and heart rate reactivity, such that they showed a dampening. Children who were low income at the beginning of the kindergarten year showed a successive dampening in their stress reactivity across the kindergarten year. Also striking was using the MacArthur ladder of social status, subjective social status, which Nancy Adler and others spearheaded many years ago, where people rank themselves relative to other in their community. How do they feel they stand in society? And what we found is, above and beyond the kind of the objective SES measures, that parents' rating of subjective social status accounted for additional dampening in children's sympathetic reactivity and heart rate reactivity, suggesting that there were these social factors that parents were experiencing that were trickling down through the child's experience in a manner that was leading to dampening or muted stress reactivity at age 5. We also find evidence for this in DNA methylation marks in the children at age 9. And interesting, we found them predominantly at loci with high relevance for mental and physical health, and in particular, loci related to immune function. We found both hypo and hypermethylation evidence in quite a few loci. And what was striking is that this is associated, for those of you guys less familiar with gene expression and function of these genes, whether or not they are likely to be turned on or off. And these effects were unique by exposure. So there was very little overlap in the loci methylation that was predicted by income, by education, or family adversity. And also important to note is that the family adversity, though many people think of it as the most prominent exposure of risk, it's really that income and education that were the strongest associations. More recently, one of our postdocs, Kristen Rudd, who works with me this year, wanted to look developmentally using very complex developmental models and also considering multiple systems. I talked about 3 different biological markers. Of course, those work together. We have very limited understanding of how they work together. She used some very sophisticated modeling to create latent classes of stress physiology at rest and in reactivity, looking across parasympathetic and sympathetic nervous systems, as well as the HPA axis. And what she has demonstrated is that exposure to early adversity in the first 18 months of life predicted what type of multi-system profile children had at age 3. And in particular, kids who experienced a lot more adversity earlier in their life were more likely to be in the anticipatory arousal profile, such that they entered the challenge and the stressful experience of our lab before we even started to stress them. They had a higher resting heart rate, higher resting cortisol, and greater sympathetic activity. And that when the challenge came about and they were supposed to mount this appropriate stress response to these developmental challenges, they had weak or moderate responses, frankly, because they may have been somewhat ceiling down at the beginning. This particular profile is associated in our data with executive function at age 5, a couple years later, providing evidence that there is this mediation through multi-system changes in stress physiology from early adversity to later functioning in a manner that, for those of you guys who are less familiar, executive function is related to obesity, behavioral and mental health, as well as a variety of other life outcomes. So quick summary of what I've shared with you so far. We had this accumulating evidence showing that early life adversity and socioeconomic status relate to and shape reactivity across multiple systems. I just highlighted a few today to give you a sampling, but that this occurs even in typically developing samples. This is not exclusive to those with extreme trauma. Moreover, we've demonstrated, and one study I showed you and others, that these biological changes early in development, age 3, age 4, age 5, these behavioral phenotypes, biobehavioral phenotypes predict risk for later health and well-being. Therefore, we really have to understand and identify these phenotypes. It's really important to be thinking about these early in life, because those of you guys, the pediatricians or any MDs or anyone who studies biology in the group understands that variation in sympathetic reactivity or glucocorticoid arousal over time can often lead to compensatory activity to try to hit allostasis. And this can contribute to disorders. There's a construct thought of as allostatic load. That's a rich literature if you want to learn more about that. And that also this can lead to different health behaviors to adapt and cope with the arousal that people experience, substance abuse, et cetera. And so I got really interested in continuing to demonstrate this as a potential pathway for SES and adversity effects that might be compelling to policymakers. I co-edited a special issue on this topic around biological mechanisms of early life stress and physical health. If you're interested in reading more, there's a nice compilation. It's a couple of years old, but has a great place to start in this literature. Importantly, not everyone who's exposed to stress or adversity has negative effects. And in fact, we vary greatly in our susceptibility to environmental exposures. And Tom Boyce, who's an emeritus professor who many of you know, championed the construct of biological sensitivity to context. And this basically is an idea that says some of us are very high in biological sensitivity. We're more affected by environmental input, whether that be good or bad, for better or worse. And those of us who are very sensitive, if we're in a stressful environment, we're going to have worse adaptation. But if we're in a more supportive, nurturing context, we may do better than our peers. In contrast, people who are less sensitive to the environment often will do equally well across both types, or at least close to equally well. And the dandelion and orchid hypothesis is one way that people have conceptualized this to help them think about the sensitive individuals being the orchids who are hard to grow unless you have the right environment, and the dandelions who rise up against all environments and lawnmowers. And indeed, we have shown in our data that individual differences in temperament, but also biological markers like HPA axis, ANS reactivity, as well as genetic risk or sensitivity alleles, all moderate the effects of the environment, such that those who are more sensitive do better in better environments and worse environments, helping us understand who may be at greatest risk for some of these adversity effects. But along my postdoc, I got very interested in pregnancy as a particular period of biological embedding. And a few months later, after I started developing a grant, it became popularized in the media with the cover of this Time magazine talking about prenatal programming. And that is because the intuitive environment provides information about stress and nutrition, tastes and toxins to the developing fetus. And this information actually directs the fetus's development. This phenomenon describes the journey from genotype to phenotype. And this is where the embryo or fetus responds to information during sensitive periods of proliferation and differentiation and maturation. And this response results in structural and functional differences in cells, tissues, and organs and set points. And these changes relate to developmental plasticity across the life course of that offspring, either independently or through interactions with processes, and confer risk for health and disease outcomes. We are starting to learn a lot more about how this occurs. We're still unpacking it, but we do know there's an enzyme in the placenta that protects the fetus from maternal cortisol, but that roughly about 10 to 20% of mom's cortisol do pass through the placenta. And this 10 to 20% accounts for about a third of the variability in fetal cortisol. So it is directly related to that developmental glucocorticoid process in the fetus. And fetal glucocorticoids, of course, play a really critical role in brain maturation. They affect neurons and synapse formation, myelination, glia and CNS vasculature. And all of this, of course, regulates fetal growth, brain development, organ maturation, all the things we care critically about in pediatrics, and affect health throughout the lifespan. And there is an increasing literature actually demonstrating that maternal stress in many ways, whether that be perceived stress, depression, anxiety, or biochemical markers such as cortisol or placental CRH, for women who have higher levels of these stress measures, their offspring are much more likely to be behaviorally and biologically reactive to stressors, and they're more likely to recover more slowly from a stress response. These offspring also have a greater risk of preterm birth, low birth weight, neurological functioning, and mental and physical health problems. And so I wanted to dig into this world at its early stages, and some of my early work worked here at UCSF with the MAMA study, which was led by Alyssa Epple, Nancy Adler, and Barbara Laraya here at UCSF. They enrolled over 200 overweight and obese women around mid-pregnancy. They were all low to middle income, and about a third were single parents. And they were only enrolled if they did not have diabetes or other conditions affecting metabolism at the time. Half were assigned to a mindfulness intervention, which I'll tell you about in a few minutes. And I took this wonderful cohort and I got my first R01 to study the effects of stress and BMI and adiposity in the women during pregnancy, and its association with offspring outcomes across a range of developmental indicators. I was able to enroll 180 babies from that first cohort, a little over 200. You'll see it's quite diverse and very representative of many communities in the Bay Area. Because of those initial restrictions on chronic health problems, we had not too many low or high birth weight babies and not too many preemies. And we were able to follow these kids through age 5. This is a picture of one of the groups of the mamas and their beautiful babies in their mama's onesies. And I just want to honor the incredible participants who gave so much of their personal freedom and life and time for this research to enable us to do this learning. These women reported a very high level of trauma and experiences of stressful life events during pregnancy. We used the CDC PRAMS indicator, and we found that the women on average reported, more than 75% reported having at least one major stressful life event type during pregnancy. And indeed, over half reported 3 or more. And some of these are quite substantial events that would cause significant and potentially very enduring distress. And what we found is that the more types of major stressful life events the woman had experienced during pregnancy, the greater her infant stress reactivity was in the parasympathetic system. So using, again, standardized paradigms that are appropriate for 6 months old, we were able to detect a pretty meaningful association with offspring stress reactivity. We also found that for each additional type of stressful life event that a woman reported during pregnancy, there was a 40% greater odds of her offspring having rapid infant weight gain in the first 6 months of life. And working with a medical student here at UCSF, Jennifer Savitz, who's now a practicing pediatrician in Seattle, we also did medical records abstraction from over 52 pediatric practices that these babies were engaged in. And we found that women who were more stressed in pregnancy had babies who were more physically ill via medical records across the first year of life. And they showed higher rates of both infectious and non-infectious illnesses. And interesting, these were not kids who were just coming in with recurrent same type of illnesses. These were kids who had a greater diversity of illness and diagnosis profiles, suggesting that this is really proliferating across a range of systems and outcomes in these kids. So I began to think specifically about this intergenerational transmission of social experience and think particularly about the female body and how it might manifest in a manner of relevance for her and her offspring. I had the great privilege of working with a cohort based in Memphis, Tennessee. PI Fran Tylafsky has led that since 2006 and now has been succeeded by Alex Mason and Kaya Lewin, who's on the faculty at UCSF, and I co-direct the science on that project for the last 8 years. And that is about 1,500 women were enrolled in second trimester. We have a phenomenal retention rate through age 8. It's about 2/3 African-American, 1/3 white, which is quite similar to the representation of the urban South, Shelby County, Memphis metro area. I worked with Iris Stein, who's a visiting scholar, to look at maternal history of childhood trauma and how her own experience of abuse in childhood or conflict, in addition to her pregnancy exposure to stressors, might relate to fetal outcomes in this larger sample with a lot of adversity. We got really interested in considering whether or not the corticotropin releasing hormone, which is synthesized and secreted by the placenta, might be a potential mechanism for some of these effects. For those of you guys who are less familiar, levels rise exponentially in the last several weeks of pregnancy. And this is a good thing. It's very critical for fetal development and onset of birth. But too much can be related to adverse developmental outcomes for the fetus and the baby, as well as for the mother postpartum. And so we were able to look at 2 time points of CRH to assess level and rise over pregnancy. And what we found is that mom's history of their own trauma during childhood, but not her stress during pregnancy, was associated with the level and rise of PCRH in these women, suggesting perhaps a sensitive period for the programming of this gestational hormone that had relevance for mom and baby. Following up on this work, worked with a postdoc, Dylan Brown, and found that childhood trauma, when we looked at repeated measures, very sophisticated modeling by Dylan and colleagues, to look at maternal psychopathology from pregnancy, age 1, 2, 3, and 4 of her offspring, as well as developmental milestones as indicated by the scales of independent behavior in those babies, who later were 4-year-olds, we were able to hold constant the stability of some of the psychopathology and developmental outcomes and try to really understand the indirect and direct effects of mom's trauma history. And what we find is that higher pre and postnatal maternal psychopathology did predict later developmental milestones, and there were multiple indirect effects linking mom's trauma history via psychopathology, but also to suggest in a pattern that there's both biological prenatal programming occurring, as well as through postnatal maternal mental health socialization of her offspring. Similarly, working with Danielle Rubinoff, who's on the faculty at UCSF here, we repeated this approach, but this time looking at specifically maternal depression and offspring internalizing symptoms. And similar to what was found by Dylan and colleagues, our paper demonstrates that there are cumulative contributions of mom's trauma history on her own psychopathology, as well as the children's internalizing symptoms. And indeed, there's certain paths that mediate statistically trauma history effects on maternal mental health, on child mental health via maternal symptoms. So one of the key things here is to really note that there's both a contribution in pregnancy and an accumulative contribution to maternal mental health and child outcomes across the first several years of life. That effect isn't a one-time effect, even though it happened many, many years ago. And this is one of the reasons that there's such a advocacy for identifying maternal depression in wild child visits. And of course, there's hardly enough time to do this and rarely enough resource to refer families to. But this is just more and more data to suggest why this is so critical if we're interested in taking care of children and their health. We looked also trying to understand, is there a specific type of stress during pregnancy that might be most predictive of these outcomes in babies? Is there something, a unique exposure that we want to understand? And so Sheikh Ahmad, who's a postdoc here at UCSF right now with us in the Division of Developmental Medicine, demonstrated in this cohort that both childhood trauma, as well as 3 different types of stress domains in pregnancy, socioeconomic stress, intimate partner violence, as well as neighborhood violent crime, all uniquely and cumulatively predicted socio-emotional well-being in those 1-year-olds born to these women. It's important to note that violent crime has one of the highest rates in our country in Memphis, Tennessee, where the sample was drawn from. And it's just really notable that these effects are above and beyond income and education and all of the important key covariates you would expect. And that collectively, these account for a lot of variability and behavior problems in these kids. We wanted to take this and say, well, is this just true in Memphis, where there's a lot of crime, a lot of stress, a lot of disparities due to Black and white histories of inequality and oppression? And so I worked together with colleagues in a multi-site study. We are MPIs on Echo Pathways study, which brings together 3 cohorts from 7 different cities across the country to understand child development from pregnancy through middle childhood. And we just finished these results this week, actually, looking both in a multi-cohort pooled analyses, which you see on the left panel, and then also breaking down these effects by cohort to just understand whether it's driven by any particular cohort. We see that both pregnancy, stressful life events, as well as a history of childhood trauma, each of them uniquely predict in unadjusted and fully adjusted and over-adjusted models behavior problems at age 5 in these kids, suggesting that we can have a lot of confidence that this is a true effect across a variety of populations. So collectively, this work demonstrates not only the biological embedding of stress across the life course, but also that these effects are transgenerational, and that a mother's own history of adversity influences the programming of her own offspring decades later, and that we have to consider children's health in the context of the family system and its history. And that's a lot of bad news, and I hate to be the bad news person. A lot of my work actually focuses on what we can do about this and for whom we can see evidence of protection, resilience, prevention, intervention, and potentially reversibility of harm from these harmful exposures. So again, we have over 40 different types of outcomes that we know ACEs are associated with, but let's take a little deeper look. This is a particular focus on a paper showing prevalence of depressed affect, showing this linear association. And on the far right, you see those with 4 or more adverse childhood experiences have the greatest risk for depression in the sample. But I want to point out that actually over half of people who have 4 ACEs are not depressed. And there's very little attention to what predicts being in that green bar. And so we actually really dig into that quite a bit in my lab. And one of the things that we look to is what allows stress to be tolerable. There's toxic stress is a hot word out in the field. It's used a lot. I have mixed feelings about it as a word. It can be very stigmatizing. But it is useful to think about how some stress is good. You want stress, it's helping me perform and provide this lecture now, but you want it to be brief and you want to feel like you're in control and that you're able to calm after the stressful encounter. Tolerable stress is serious. It has temporary stress responses and is potentially buffered by supportive relationships, whereas toxic stress is thought to be prolonged activation of stress systems in the absence of protective relationships that might buffer or protect people from those feelings and sensations and bodily responses. So we wanted to identify malleable protective factors that would buffer maternal stress effects on offspring health. And we dug into our data sets to see what we could use to test these questions. And Dr. Ahmad's work has found some really interesting and promising things, and this work is under review. In particular, there's a strong association between socioeconomic stress in pregnancy and children's socio-emotional behavioral problems. But what Dr. Ahmad found is that this is moderated by maternal knowledge of infant development, her awareness of developmentally appropriate expectations for her baby and the ways in which she might respond developmentally appropriately to their needs. And what we find is for moms who are low in this knowledge, there's a strong association between socioeconomic stress in pregnancy and child outcome. But for women who have, one standard deviation or higher on knowledge of infant development, there is no association, and those kids are buffered from that potential risk of stress. Similarly, looking at age 4 outcomes, externalizing behavioral problems as well as executive function, we found a similar pattern, this time using objectively coded parent-child interactions, where we coded parent behavior for warmth, responsiveness, cognitive flexibility, all of the good things you want to see in our interaction with the child. And we did this repeated over time in the first couple of years of life. And similarly, that red line is the association between stressful life events in pregnancy and child externalizing on the left and executive function on the right. And you see a robust association for those who are low in parenting quality. But for those who have high parenting quality, that association is completely buffered. So again, evidence that this postnatal malleable environmental factor can really buffer prenatal stress effects on offspring outcomes. We found similar types of story looking at stress physiology. Karen Jones-Mason and Michael Kosia in the lab here at UCSF found that for those who had low quality parenting, there was a robust association between stressful life events and infants' sympathetic nervous system reactivity at 6 months of age. But for women who demonstrated a high warmth and responsivity to their children and were able to regulate their infant's distress during the challenge, and there was no association between her stress and pregnancy and sympathetic reactivity in those babies. Similarly, low quality parent-child relationships with a lot of negativity. For kids who have that kind of caregiving relationship, there was a robust association in the PAUSE study between socioeconomic status and health impairment in kids in kindergarten. Whereas if there was a low negativity in the parent-child relationship, there was no association between socioeconomic status and health impairment. So again, that quality caregiving relationship buffering and protective. But it's not just parents, there's also macro level societal factors that make a big difference and can be quite impactful. Danielle Rubinov's work here showed that this objectively measured neighborhood quality, measuring a range of features in the environment that we linked geospatially to our participants, that socioeconomic status has a robust relationship between SES and physical health in the spring of kindergarten, adjusting for fall health, but that for children who had the good fortune of living in a neighborhood that was very high quality with a lot of opportunities for education, childcare, healthy parks, and a variety of other positive factors, healthy food, et cetera, there was no association between individual family SES and physical health changes over the kindergarten year. So with all this good news, we got excited. Like, can we not just capitalize on positive things that already exist in the environment, but could we potentially prenatally program a positive outcome for offspring? And again, circling back to that MAMA study I told you about, working with Alyssa Eppel and others who found that women who participated in the mindfulness-based stress reduction 8-week intervention, had a substantial decrease in perceived stress and depression from the beginning to the end of pregnancy relative to those who were in the treatment as usual group. And importantly, work by Jennifer Felder, who was a postdoc and is now on the faculty here at UCSF, she demonstrated that women in the treatment as usual group, these effects were sustained. So women in the treatment as usual had almost an 8-time greater odds of being in a moderate, stable depression class through 18 months postpartum relative to women who had experienced the mindful health promotion intervention in pregnancy. So again, we have this big difference in intervention in maternal mental health in pregnancy, but also postpartum. I want you to keep that in mind because that depression at 18 months in those women predicted concurrent child internalizing problems and also predicted additional changes and increases in those children's internalizing at age 4. And so affecting that mental health has this postnatal impact on well-being on these kids quite a bit. And also work with Amanda Neroni-Zhu, who's a staff member in our Division of Developmental Medicine. She helped us write up the results to find that babies born to the women in intervention also demonstrated faster sympathetic reactivity to the stressor, which is good. You want a fast sympathetic response to challenge. And overall, they recovered fully, had no difference between baseline and the recovery period and sympathetic reactivity, whereas kids in the treatment as usual group had no response initially when they should have, and then a delayed sympathetic response to the challenge, and then did not fully recover at the end of the task. We coded infant behavior. We found babies in the intervention had more self-regulatory behavior throughout the task and more negativity throughout the task, suggesting both temperamental and behavioral evidence as well as the physiological. Thinking specifically about some things that pediatricians think a lot about is infant healthcare utilization over the first year of life. And we were really surprised and interested to note that participation in the mindfulness intervention also had effect on infant healthcare utilization. So for women who ended up in that moderate stable depression class, which again were 8 times more likely if you were in treatment as usual, if you were in that class and you had received the intervention, you were more likely to attend a more normative number of healthcare utilization visits for your infant. Whereas treatment as usual, mothers had an additional 6 visits per year for their infant. And our data suggests that this is both because the infants were more sick, but also because women who are depressed tend to seek healthcare a little more often, and this suggests they may seek it more for their offspring as well. So one of the things I hope you will take away so far is that there's multifinality. Early life trauma, adversity, no matter when it occurs in development, can lead to a lot of different outcomes, a lot of biobehavioral outcomes, but a lot of physical and mental health outcomes. And there's also a lot of diversity in who has a negative outcome. So a paper that just came out this year that is of great importance that I hope everyone will be paying attention to is that there's a big difference between population averages in terms of risk after adverse childhood exposure and individual outcomes. A very large study conducted by very thoughtful, rigorous people found that children who had more ACEs had a greater risk of mental health and physical health problems at 45 years of age. So that's consistent with a lot of the other evidence from the Validity work. But importantly, they did very sophisticated analyses to look at individuals and whether or not understanding their ACEs count helped predict an individual's specific likelihood of illness. And what they found is ACEs were actually very inaccurate, poor accuracy at predicting which children would have mental or physical health problems at age 45. So it's good at a population level, but just knowing that there's a child who has an ACE in your office, you actually are not going to be very good at predicting the level of impact on their biology and subsequent mental and physical health. And this is really important. And one of the reasons that screening alone for adversity is not going to be enough. And of course, it's easier to build strong children than repair broken people. I'll help Mr. Douglas out there. And it's good to prevent, but sometimes we cannot prevent, and sometimes people have been harmed. And so I've also had the great privilege of working with some of the most amazing interventionists in the world. In particular, worked with Alicia Lieberman and Patricia Van Horn, who developed child-parent psychotherapy, which is one of the most effective and best known interventions for trauma exposure in early childhood. They have 5 randomized controlled trials across 2 different sites and programs, which have shown independently that it is highly successful at improving child outcomes, maternal outcomes, caregiver outcomes, and the parent-child relationship. So I leveraged this incredible intervention and infrastructure of research here at UCSF and partnered with them and others to understand the effects of early life trauma on maternal and child physiological systems, as well as their physical health outcomes. And our goal was also to demonstrate how there may be biological improvement from the intervention rather than just this mental health and functioning outcome. So we enrolled 59 families in this intensive therapeutic intervention. It's a very diverse and quite low income community, and they have experienced a lot of different major life adversities and traumas. The mothers on average had 12 different types of major events, and the children aged 2 to 6 on average had at least 5. So very, very traumatized population seeking therapy at UCSF San Francisco General. Worked with 2 phenomenal teams at ZSFGH and BCH Oakland to do clinical assessment and biospecimen collection at baseline. And then after about 20 sessions of child-parent psychotherapy, which included weekly dyadic treatment sessions following that assessment. We were thrilled to see that just like in all the RCTs, our population of 59 had incredible improvement in children's executive function, which is a task-based measure, as well as their mental health. And similarly, the caregivers improved substantially in all of their mental health outcomes and in their parenting quality. And we decided to pick one of UCSF's favorite biomarkers. Our Nobel Prize winners here helped understand the process of telomere length and telomerase. And for those who are less familiar, telomeres are the DNA protein complexes that cap chromosomal ends of cells, and it confers stability to the chromosome. And what happens over time is that cells divide, and as they divide, telomeres shorten, and eventually cell division stops. It's important to understand that this is associated not only with trauma leading to faster shortening and shorter telomeres in childhood and adulthood, but that shorter telomeres are also associated with depression, Alzheimer's, cancer, diabetes, cardiovascular disease, etc. And that in general, inflammation is produced in excess by these senescent cells, which is one pathway for systemic inflammation and disease. So I leveraged the Bay Area seed cohort I told you about, and we used propensity score matching to create an ethical comparison group for our traumatized sample. And in this matched sample, we were thrilled to see that the groups did not differ in telomere length at baseline. And what you see is a rapid decline in telomere length on average, which is expected in the first 5 years of life. The attrition is most significant in the first 5 years of life and slows somewhat, but continues throughout the life course. What you'll notice is the purple is the community sample. So they are showing an expected rapid attrition in telomere length over time. And the orangish red is the child-parent psychotherapy intervention. You see a lot more variability, and on average, you see what looks like kind of a flat slope. And indeed, when we test this statistically, we see a statistically significant difference between the two groups, such that the community sample actually shortened on average about 500 some base pairs per year. That's a little bit more than would be expected for an average child, but because there's such a high stress matched sample on low income and high adversity, this rapid shortening is expected, whereas in the intervention group, you see no significant shortening or change in telomere length on average across that sample, suggesting that the intervention stopped cellular aging to some degree. And exciting to think about the fact that many of these slopes are actually pointed upwards, suggesting potential lengthening. And indeed, our data show the odds ratio of lengthening is about 4, or in adjusted models 3.25, suggesting the potential for reversibility of trauma-related telomere shortening in the children who experienced this phenomenal therapy. And clinical practice is amazing, of course, but we try to think about this stuff at scale. How can we think about this in a public health framework so we can impact more than one child or family at a time? Because the whole society is really where we need to intervene. I hope you've gleaned from my narrative that there's common paths, there's common mechanisms through developing biology that cross generations, and they all move towards a broad range of health outcomes that many of us care about. And our work in silos often makes it challenging to notice and discover these commonalities, and that's been a priority of my work. We really are trying to argue in our group that we need to spend more time addressing the root causes of systemic changes in biobehavioral phenotypes that place infants and children at risk for later development of health problems, rather than waiting for the disease to manifest, and that we need to think about this at a population level. These aren't just experimental samples of 59 or 100 or 3,000, but that this is something occurring across populations. And I've been working with a pediatric innovation initiative led by the Center for the Developing Child at Harvard, where we're trying to develop a battery of biological and behavioral measures that we could implement within pediatric primary care that would help us identify which children are most sensitive to adversity. So beyond just ACEs count, how can we figure out who is evidencing a response under the skin that suggests they need a little bit more attention before overt problems appear? And we're also trying to measure stress effects as well as resilience and core capacities so that we can respond in a way that could be useful with emotions that are sensitive. And this partnership is also very, very strongly based in community partners to ensure that these measures are really sensitive and used to empower patients and promote well-being and resilience with a lot of sensitivity to the communities the data are drawn from. And speaking of sensitivity, there's so much mother blame in our society, and a lot of the data I presented you, could be interpreted in that light. She should have had a different diet. She shouldn't have been so stressed. She should have exercised more, slept more, etc. Most of the things, she should have been a better parent or known more about development, and then she could have been that green line in Nikki's slopes. Well, the problem is, of course, that most of this is out of women's control. And more important than identifying and supporting those individuals who are at risk is that we need to eradicate or resolve the fundamental causes of disparities in health. And in particular, we need to call out the harmful effects of income inequality, which are massive, of racism, and of gender inequality. Gender inequality sometimes occurs in the form of violence against women. We've talked about interpersonal violence in my data, but also on unequal opportunities and lack of support for new parents. And so focusing on this thing will help us actually prevent unnecessary harm rather than talking about what we can do to buffer the impact of these adversities. I'm also proud to be among many leaders involved in the NIH-funded ECHO National Consortium. We are collectively for the last 5 years and have several years ahead trying to gather data on over 50,000 families that are focused on 5 primary pediatric outcomes that have longstanding public health impact. And I've also been very proud to be one of the founding members of the DEI working group on that national consortium, really pushing to promote issues of diversity, equity, and inclusion, both in the study design and the interpretation and representation in this amazing consortium and the data that it collects and disseminates. And these data are collected from many sites around the United States. My sites are the ones in red, and it's such a privilege to co-direct that work with so many amazing scientists from many different fields, many different disciplines, which is helping us be more thoughtful. I recently published something with a lot of colleagues from ECHO outlining the role of the family environment in neurodevelopmental risk and talking about the opportunities in the ECHO study, which explains some of the measurement and the plans for those who want to know more. These data are going to become publicly accessible quite soon. And so those who are interested in any of those pediatric outcomes who would like to use ECHO data to conduct their own inquiries should start learning more about ECHO so that they can be prepared when those data come online. One of the studies that I'm an MPI on within the ECHO consortium is the ECHO Pathways study. I showed you some findings from that earlier. What I wanted to highlight is that it just gives us a lot of opportunities to do, transdisciplinary work. We're looking at chemical exposures, both phthalates and air pollution, in addition to the social stress exposures that I highlighted today and their interaction to predict both neurodevelopment and airway outcomes. We also have papers looking at obesity, premature birth, blood pressure, et cetera. We have a lot of different papers underway. We're also looking at placental transcriptome as a potential mechanism with a lot of attention to sex-specific associations. And I'm also a site PI on the NYU Pediatric Obesity, Metabolism, and Kidney Cohort Center, where we're looking at synthetic chemical exposures in utero and childhood to understand their influence on developmental metabolic programming. So that one is also underway and has a lot of exciting data to share, perhaps in another talk. So what, why I rushed through a ton of different information, but I think there's a lot to be gleaned here for pediatrics. There's a lot that we can do about it. Often we feel really taxed. We've been hearing about toxic stress for over a decade and how critical it is to translate this into our practices and into our care and our thinking and our research. And we've made some progress, and in particular, UCSF has led many ways nationally in making sure that we are doing these things. There is a lot of recommendations around screening for depression, but as I noted, it's only in the perinatal period, and it clearly matters longer than that. UCSF has led in screening for social determinants of health, both in the adult medical care, but also in pediatrics and emergency care. And some amazing leaders at UCSF, Laura Gottlieb and Dana Long in particular, I'm familiar with, who are screening for social needs and doing something about them. And then of course, focusing on family strengths so that we can amplify those and support them. Many are aware of the ACEs Aware movement. We are so privileged to be in California, where our governor and Surgeon General prioritize this issue and have pledged to cut ACEs in half. And California is leading the way by training and providing payment to Medi-Cal providers for ACEs screening. Many people here at UCSF are leading this work and incredible champions for this incredible task. And there's a lot going on that is incredibly exciting. And so there are solutions, coming about. There's a lot of learning to be done. And we frankly need a lot more data. As I noted, screening for ACEs alone won't work. So we're trying to identify the many different ways we can use that and complement with other information over time to refine a precision medicine approach to identifying risk and working with families. And Rafi mentioned in his introduction that I'm an MPI on a recently awarded project from the California Initiative to Advance Precision Medicine with Nita Thakur and Dana Long and Danielle Hessler from UCSF, as well as our community partner PI, Lisa James from Futures Without Violence, and our San Francisco PI, Dr. Kia Keating, as well as many co-investigators from UCSF, to really try to draw upon a primary care-based identification and provision of intervention with a strong consideration of stress biology and community partnership to translate that into effective solutions for families experiencing challenges. Hopefully I'll have some data with my colleagues to share with you in the coming years. And I guess I'll end with saying, healthcare can be social care, and we need to both be aware, but also offer assistance and make adjustments. And that pediatricians, really, we have an opportunity, psychologists, to align with these communities, to advocate for policy change so that we can get payment for the things that actually affect health, not just wait until someone is ill, and that social needs are health needs, and that the more we can do to get the policies and practices to align with what is true in the data and what we know in our hearts, the better off we'll be. And then finally, just to comment that, of course, we can't just make sure everyone has equal access. We need to recognize that there's been centuries of inequity that have led to huge disparities in biological, mental, and physical well-being, socioeconomic well-being, and we need to do something in our policies to make sure that that gap is closed. And I just want to end by saying I have intense gratitude for all of you listening. You're in pediatrics, not because of the money or because being at UCSF is easy, but you're here because you know that the kids are where it's at. And I just want to recognize you and honor you for the work that you do. And thank you, because there is no keener revelation of a society's soul than the way in which it treats its children. And finally, just want to acknowledge the incredible people who helped me do this work, both my mentors and colleagues and co-investigators, and the phenomenal lab team pre and post pandemic who get this work done with me and for me, and many colleagues at UCSF and outside of UCSF who enable this incredible data and interpretation I've shared with you today.
Thank you, Nikki. That was really amazing. Such impressive work and so important as well. So want to compliment you and also thank you for all you've done in this such an important topic. Let me try to summarize maybe some of the questions and comments that have come in to shoot back at you with your thoughts. So one seems to be a few questions related to for a pediatrician in a busy practice. I guess two-part question. One, are there alternatives to doing ACEs screening that might be easier for a busy pediatrician to tackle, number one? And then related to that, if the screening shows a concern, what's the next step that the pediatrician can do practically for their patients?
So thank you for the question. I both love and hate it. It's the challenge that we talk about every day in this work. We don't want to criticize the current screening because we need to increase awareness. We need to consider who may be at risk, but there is a lot of stigmatization that's happening. People presuming that someone with adversity is harmed or damaged in some way, or that they want support or intervention. And so it's a tension. And I think what we're working really hard in partnership with providers and practitioners who are on the ground with little time to do this stuff is to identify biological panels, biobehavioral panels that can be implemented not by the doc, but by MAs or others who we can use in conjunction with the questionnaire data, just like you might a metabolic panel or lead screening, to better target who may be in need and more responsive to additional resources. And then, of course, it helps if people have strong training and trauma-informed care in early training, so that the stuff isn't so awkward and intimidating and time-consuming when you are in practice. And so I think a lot of those things are under development, but we're not there yet. So I don't have a great answer that will make people feel relieved, other than there's a lot of people trying to address that challenge.
Great. And another question kind of related is, so the really nice results you showed with your intervention study, the question is whether those services and those interventions are still available at ZSFG, at Oakland, at any of the other sites.
Yeah, they are. And I think the teams at Alicia Lieberman's Child Trauma Research Program, they provide and it has been free for many, many decades. So I don't know if it is today, but it should probably be. It's very, very dependent on philanthropy. But that is accessible to many people, so you can reach out to them. And also in Oakland, that will depend on the payer mix and whether or not you're eligible in that particular clinic. Early intervention services provides that there. But there's many, many programs throughout the Bay Area that provide child-parent psychotherapy, because there's been incredible support by the Harris Foundation, as well as Lisa Stone Pritzker and others, to make sure that this is available for many people outside of UCSF.
Okay, I think I'll try one more question. This is a bit of a technical question, Nikki, but
okay.
The question is, why did some of the curves inflect upwards for the high SES group in your intervention slides? Was this a statistical aberration, or is it something significant?
I think the only curve was not an intervention slide. The curve was in a community sample of 338 kids. And we think that curve is related to the fact that some stress response could be good or adaptive. A moderate amount of arousal does lead to better test performance and school function. And so that may be what that's illustrating. Alternatively, it could be that those wealthy kids are in really stressful school environments, because we did collect that data at the beginning and end of the school day. There's no way for us to know with those data, but we do have those two alternate hypotheses of why that might be happening.
Okay, great. Well, I want to thank you again for such a fantastic grand rounds. Thanks to you, and thanks to the audience for participating, and wish everybody a good rest of your day.
Thank you for listening. Such a privilege to share this work with you.
Thanks.